Hello, my name is Matt Kenworthy, and I am a postdoctoral fellow with the NOAA Living Marine Resources Cooperative Science Center, and I am located in Savannah State University. The project I am sharing with you today is a study conducted by myself and my supervisor, Dr. Dion Hoskins Brown. Today I will be sharing with you the details of a study we are working on evaluating the effects of landscape scale habitat variability on white shrimp population dynamics in Georgia estuaries. I think we all understand and respect that coastal habitats provide a variety of ecosystem services, including shoreline protection, erosion control, nutrient cycling, carbon sequestration, and provision of habitat for many commercially and recreationally important species. Unfortunately, these habitats are declining at alarming rates, leading researchers and managers to further identify the value of these habitats and the ecosystem services they provide. In this talk, I will be focusing on the value of salt marshes and tidal creeks as habitat for white shrimp in Georgia. When we think about species-habitat associations, there is a common perception that more habitat equals more fish and crustaceans. Classic studies have shown this close relationship to be true. In the graph on the right, you can see results from Turner 1977 that demonstrated that shrimp catches in the northern Gulf of Mexico were highly correlated with nearby marsh area. While this is a strong example of high correlation between habitat availability and population size, species-habitat associations are not always this clear. Understanding the value and function of estuarine habitats as habitat for fauna is quite challenging. These associations are complicated by a number of factors, including spatiotemporal variability in animal presence within target locations, concerns with scaling up from a small number of observations to the entire estuarine system. Animals behave individually. Estuarine ecosystems are also complex mixtures of a variety of habitats, and additionally, individuals move. Considering these challenges and the complicated nature of estuarine ecosystems, we need more quantitative analysis of species-habitat associations. Before getting into the details of this study, I wanted to touch on the nursery function of estuarine habitats. Nurseries for marine fish and crustaceans are locations where individuals spend their juvenile life history stage foraging and growing until they recruit to adult populations. In 2001, Beck et al. Introduced the nursery role hypothesis, which suggests that the value of a nursery is based on the per unit area contribution to the production of individuals that recruit to adult populations. According to this hypothesis, if habitat A is the size of a basketball court and habitat B is the size of a football field, and both contribute the same number of individuals to the adult population, then habitat A, the smaller habitat, is of higher value. However, in 2015, Nagelkirchen et al. Expressed that the estuaries are spatially explicit units, or seascapes, consisting of functionally connected mosaics of habitats, incorporating ecological processes, and that we must consider how those habitats are arranged and the characteristics of the habitats when we evaluate their value and function in supporting populations of marine organisms. It is this train of thought that we are considering in this study to evaluate the value of estuarine habitats in supporting white shrimp populations in Georgia. White shrimp are important from both an ecological perspective in that they are an important prey resource in the ecological food web, and an economical perspective in that they are a target species in a multi-million-dollar fishery. White shrimp are also harvested as bait shrimp to be sold to recreational fishermen and women. It is well known that white shrimp utilize tidal creeks and saltmarsh habitat as juveniles to forage and grow. However, there has been limited amount of research evaluating whether all tidal creeks and salt marshes are equal in contributing to adult populations, or if there are certain areas within the estuary or certain characteristics of the tidal creeks and salt marshes that are more valuable for the abundance, growth, and survival of individual shrimp. For example, Ron Kneib's research from Georgia some years ago suggests that landscape characteristics such as marsh continuity and marsh platform elevation can influence shrimp population dynamics. Now, before moving forward, I want to say here that this study is a collaborative effort of many individuals, including co-PI Dr. Dion Hoskins Brown, NOAA collaborators Jennifer Dorr and Jennifer Leo, Mark Fisher at the Skidaway Institute of Oceanography, and multiple students, including Cameron Atkinson, William Burns, Maya Clark, and Kaylin Guinness. In this talk, I will be covering three main themes or questions. One, how does shrimp abundance vary spatially? Two, how does shrimp condition vary spatially? And three, how does tidal creek and marsh habitat characteristics influence the abundance and condition of shrimp? To address these questions, we have implemented a monthly survey of multiple tidal creeks in the Wassaw Sound Estuary. The Wassaw Sound Estuary is located near Savannah, Georgia. Within this estuary, we have identified three general locations spread out across the estuary. These locations are identified by the red circles on this map. Within each of these locations, we selected three tidal creeks that were characterized by different marsh platform elevations. We chose to evaluate different marsh platform elevations because this is a relative measure of tidal inundation time on the marsh platform, which can influence access to food resources by white shrimp. We used LiDAR data to identify low, medium, and high marsh platform elevations. You can see examples of this in the insert in the upper right corner of this map. If you are wondering about the orange circles and the yellow stars on this map, I will return to this later in the talk. For our monthly surveys, we used trawling and cast nets as our primary collection gears. We quantified the catch per unit effort within each region and marsh platform combination. Additionally, we quantified the Fulton's K condition index for individuals caught within creeks for each region and marsh platform combination. Fulton's K is quantified for each individual by dividing weight by length cubed and multiplying this value by a constant. In this case, the constant was 10 raised to the 5th power. Viewing some of our results from the trawl catch data, shown on this graph on the left is mean catch per unit effort, or CPUE, on the y-axis and the three regions on the x-axis. Additionally, marsh platform is color-coded within each region. These data suggest that there is an effect of region and month on the mean shrimp catch per unit effort, or CPUE, but no apparent effect of marsh platform elevation on these catches. Overall, mean CPUE was greater in the McQueen region compared to the Half Moon and MSRC regions. I will note that these patterns are driven primarily by catches in the McQueen region high marsh platform. Otherwise, catches are relatively comparable across all sites. Mean CPUE tended to be lowest on average in the low marsh platform elevation locations, although the results from this analysis were not significant. There also appears to be some variability in the catch data over time, which we will explore further. If we look at the castnet data, again in this graph, mean CPUE is shown on the y-axis and the three regions on the x-axis with marsh platform elevation classification color-coded for each region. The data suggests that there is an effect of region and month as well as marsh platform elevation on the mean shrimp CPUE for the CastNet data. Averaged across all marsh platform elevations, mean CPUE is shown to be greater in the McQueen region, followed by MSRC and then Half Moon. The analysis suggests that the medium marsh platform elevation locations had greater catches than low and high marsh platform elevation locations. However, this pattern is strongly influenced by the medium marsh platform elevation creek in the MSRC region. Aside from this observation, mean catch per unit effort on average again tended to be lower in the low marsh platform elevation creeks. Moving on to the condition indices, if we look at the graph on the left, which is displaying the Fulton's condition index on the y-axis and the regions on the x-axis, with marsh platform elevation classification color-coded for each region, the data suggests that there is an effect of region, month, and marsh platform elevation on the condition indices. These data suggest that on average, condition of individual shrimp is greater in the Half Moon region. Additionally, condition indices trended higher at the medium and low marsh platform elevation creeks, although this pattern was less pronounced in the MSRC region. If you recall, catch data suggested that mean CPUE for shrimp was typically lower in the low marsh platform elevation creeks, whereas here it appears that individuals in these low and medium marsh platform elevation creeks have greater condition index values. This could potentially mean that there are less individuals. This could potentially mean that if there are less individuals, then shrimp are able to grow more efficiently, or that they are potentially able to access more food resources on the marsh platform in these locations where the platform is inundated more during high tides. We also saw some effects of time in this analysis which we will explore further. So far, I have been only talking about marsh platform elevation. We also want to consider the effects of marsh continuity and patchiness, as well as the abundance of specific habitat classifications on catch data and condition. For example, the amount of oyster habitat or total area of intertidal mud banks may influence catches and condition of shrimp. To explore this further, this spring we will be conducting drone surveys to quantify these additional habitat classifications and measure marsh patchiness. We will collect high-resolution imagery at each of our 9 creeks, quantify these habitat metrics, and use them to further explain catch and condition data as well as potential implications on food web dynamics. I will talk more about food web information in just a moment. Additionally, when examining the value of nursery habitats, it is important to understand how habitat characteristics affect growth. 2 common ways to quantify growth are tag and recapture studies, which quantify growth from length measurements on recaptured individuals, and caging studies, which monitor growth of individuals kept in enclosures in the target locations. While these approaches are highly valuable, they have their challenges. For example, tag and recapture studies rely on intensive field sampling programs and are ultimately dependent on recapturing a tagged individual to obtain growth data. Further, caging studies are good in low energy and low tidal range environments, which we do not have here in Georgia. Therefore, we are testing an approach which utilizes RNA to DNA ratios as a growth proxy. The theory here is that RNA concentrations are assumed to vary with growth and condition of individual shrimp, whereas DNA concentrations are relatively constant. Therefore, individuals with greater RNA to DNA ratios are assumed to have greater growth and be better conditioned. While we plan to apply this thinking to shrimp from our different creeks, we are first conducting a lab study to test the theory. A couple of undergraduate students are exploring this by holding shrimp in lab mesocosms for a period of time, measuring the growth of each individual, and then quantifying the DNA and RNA concentrations from these individuals. From this study, we can directly compare measured growth with the RNA to DNA ratios for the lab-grown shrimp. This project is underway and final analysis of this data is coming very soon. Last on our upcoming analysis list is further exploring what areas of the estuary are contributing most to the adult population of white shrimp. To accomplish this, we are using stable isotope signatures to identify putative nursery areas. Within each of our regular sampling locations, as well as 2 additional regions of the estuary, highlighted by the orange circles on this map, we are quantifying carbon-13, nitrogen-15, and sulfur-34 isotope signatures from juvenile shrimp. Additionally, we will quantify the same isotope signatures from individuals emigrating from the estuary. These will occur at the yellow star locations on this map. From this data, we aim to match signatures from the emigrating adults with signatures identified from the different respective juvenile sites to quantify the relative contribution of individuals from these juvenile sites to the emigrating adult population. This idea has been tested and applied within estuaries in Australia. However, this has not been explored in depth within southeastern United States estuaries. Additionally, we will use the stable isotope information to evaluate food web dynamics and resource use across the different creeks and marsh landscape characteristics. To summarize our findings, the catch data suggests that there are some differences in catch per unit effort among the various regions of the estuary, and while there is some suggestion of an effect of marsh platform elevation on CPUE, these patterns remain somewhat equivocal and require further analysis. However, these data do suggest that marsh platform does have some effect on the condition of shrimp, which could be related to density of shrimp within respective locations or access to food resources on the marsh platform. We hope to provide more insight to this with the stable isotope data. These differences in condition, however, could have implications on the growth and survivorship of individuals and their recruitment to adult populations. Hopefully, we can provide more insight into this with more information on putative nursery areas. Finally, collectively, all of the data associated with this study will help us better understand the spatial variability and population dynamics of shrimp and provide information valuable for the management of this species and the habitats they use. With that, I thank you for attending this talk, and I will welcome any questions.